Welcome to the 2016 NASA Ames Summer Series. Life can be considered as an expanding, complex, dependent set of chemical reactions that once began, has optimized its capabilities to occupy various environments. Today's talk is entitled Subsurface Astrobiology, Cave Habitats on Earth, Mars, and Beyond, and will be given by Dr. Penny Boston. Dr. Boston is, as of May 2016, the director of the NASA Astrobiology Institute. Prior to that, she served as an associate director of the National Cave and Karst Research Institute and professor and chair of Earth and Environmental Sciences Department at New Mexico Institute of Mining and Technology. She received a Bachelor's of Science in Microbiology, Geology, and Psychology. Wow. Then a Master's of Science in Microbiology and Atmospheric Chemistry for the interns, look at diversity, and followed with a PhD from the University of Colorado Boulder. She has numerous publications and awards, too many for me to mention here. Please join me in welcoming Dr. Penny Boston. Thank you.
So it's lovely to have the opportunity to talk to you about some of my favorite things, and some of my favorite things are caves. I started working in caves about 25 years ago after already being in mid-career and working in other extreme environments. And I was not a caver recreationally. A lot of folks who find their way into cave science do it because they are, sport cavers and explorers and stuff, and they really want to run with that theme and figure out how to make a living out of it. I'd been in the usual number of caves as a kid, show caves, and I thought they were really cool, but I had not connected those to my science. And it wasn't until in the early 1990s I wrote a paper with several colleagues, one of whom is here, Chris McKay, suggesting that the last best place to look for life that was still alive on Mars would not be on the surface where the conditions were really, really tough, but actually in the subsurface. And so that got us thinking about how we could get into the subsurface. And we were still young and not all that well endowed with funding research. And so the idea of drilling was compelling, but it was really expensive. And we couldn't get any of the old established guys to sort of let us get our foot in the door. And so we saw a National Geographic special about this really amazing cave in New Mexico called Lechuguilla. And this is the deepest cave in North America. It is an enormous system and has currently about 140-something miles of map passage. It's a gigantic maze system. And in this National Geographic special, they said a couple of really important things. One is that it was a pristine system that had to be dug into. So that meant to us that perhaps there would be minimal human contamination in the system. The second thing was that it had all this exotic sulfur mineralogy. And for those of you who know anything about Mars, Mars is a pretty sulfur-rich planet. We've known that ever since the Viking missions. And then the third thing was that there was a USGS scientist named Kim Cunningham who was on camera and said that he thought he had seen what he thought were microorganisms in his scanning electron micrographs of some of the cave decorations in there. So I called him up and I said, hi, Dr. Cunningham, you don't know us, but we're from NASA and we'd like to go into your cave. And he said, okay, so cool. He was a boundless enthusiasm kind of guy. And a lifelong caver, which was an asset but also a detriment because he didn't understand how very hard that would be for those of us who were not cavers. And so we trained for all of 3 hours in vertical work in Boulder, Colorado, where I was then living. And we went down to Lechuguilla Cave and went in for a 5-day expedition. And it nearly killed us. And the whole time I was in there, I just kept thinking, all I have to do is live long enough to get out of this bloody place, and then I never have to come back. And so that was an inauspicious beginning to my caving career. But what I discovered when I got out of the cave was all the bruises and the eye that had swelled shut from stuff getting in it and the busted ankle and all that stuff started to heal. And then what we do as humans is kind of remodel the past, right? This is why we have more than one child usually. And so I began to forget about the bruises and the pain and the stress and how I wasn't even slightly up to the task of being in this environment. And what I remembered was what I had seen. And what I had seen was the most exciting set of environments that I'd ever seen in my life. And each room was different. And this was an amazing environment, unlike anything that I had ever seen before. And that's really what got me started. And I refocused a great deal of my work in that direction. So that's really the answer to this. Why would you go into these places that are working hard to try to kill you? Because it seems glamorous, but really it's a whole lot of no fun. I mean, it's really a lot of work and a lot of stress to try to keep ourselves safe. So I always look goofy because we're in environments where we have to actually protect ourselves, right? So all of the glamour shots that you see here are me in one kind of protective garment or another, or just liberally covered with mud, because that's the other part of it. You have to really like getting dirty in order to operate in these environments. So that's the fun part, is playing with mud pies. But the serious part is that it's very difficult to do microbiology in such a dirty environment. And so we've had to really learn how to alter our techniques to actually cope with that. Well, so that's been a lot of my gig for the last quarter century, and of course I've taught and done research in different institutions and so forth. The new gig is really pretty different in a way, but it's informed by my feel for what it's like to get out into natural environments and interrogate them. And so I have great respect for the astrobiology community, which is an enormously diverse community. So the new gig is basically this. We are housed here at Ames, and we are a virtual institute to do with all of these kinds of questions that you see up here. We're part of the overall NASA astrobiology program, which is managed and run and directed from headquarters. Like all NASA things, right, you can't get away from org charts. So this sort of shows how the Science Mission Directorate is kind of the overall lovely and happy unicorns and sparkles rainbow over everything. And then the Planetary Sciences Division is nested within that, and astrobiology in turn is nested within that. And all of those little green fruits hanging off are parts of the astrobiology program that are run out of headquarters. And then, of course, NAI, because we are the sparkly galactic center of the universe, we are here. It's been fun to get used to being queen of NAI, and it's quite an enterprise. There's over 600 different individuals currently involved in the program. This is a mix of senior science folks, postdocs, students, and a whole bunch of different lead institutions. Excuse me. The lead institutions only are the primary places where the different teams are run out of, but of course it involves a lot of other different institutions. And so this is a very exciting intellectual environment to operate in. So when I think about astrobiology, there are fundamental questions that we have, and excuse me, there are fundamental questions that we have. And one of the ones that was quite apparent to people very early on in the space age is illustrated in this really classic cartoon from the New Yorker, which appeared in 1962. Already the press kind of understood pretty well what part of our job is, and that is to think about life forms from first principles. What is life? What does that mean? What are the implications for that, for actually looking for organisms on other planets when you have no idea what they're going to be like? And so in a remarkable turnaround, this has been one of the most instructive things that we could have done for understanding life on Earth, because it caused us to really think in a much more fundamental way about how life operated here. And it stimulated people to go to the ends of the Earth, both physically, chemically, thermally, to try to see what life here could actually do. So astrobiology in general, within the framework of NASA's conception of it, really has three major arenas that it is focusing on, these three fundamental questions. What is the very origin of life, and how does it then evolve in a planetary context? Planets give rise to life. They have here, in our case, and we work under the uber assumption that this is true broadly throughout the galaxy and undoubtedly beyond. Does life exist elsewhere in the universe, and how do we go about looking for it, and how do we recognize it when we find it? And in fact, there are really fairly philosophical implications of this, which involve what the future of life on Earth is and beyond. So if you really look at astrobiology, it's not a fundamental core science, it is a place-based and problem-based science. And it really involves all of these sciences, particularly a really large dose of engineering, because we need missions to actually go to places where we want to look for life. We need instrumentation that can allow us to detect things that are not obvious. If the life were big and were going to bite us on the ankle like it does in a lot of science fiction movies, it wouldn't be such a hard problem. But we think primarily that we are looking for microorganisms, which we have a hard enough time studying here. The biology and the environmental aspects of extreme environments are, of course, encompass a lot of my work and that of many other people in the community. And then, of course, all of this is highly informed by the entire spectacular sweep of astronomy, everything from the creation in stellar nucleosynthesis of the elements that go to make up our kind of life and perhaps other kinds of life, all the way to how do we get the formation of solar systems, how do we get planets, what does that mean, what kind of planets do we get, what are their endowments of riches that they can bring as some of them undoubtedly develop into life-bearing planets. Well, there is a fundamental similarity between astrobiology in that regard and the study of caves, which is called speleology, in that it is also a collaboration of all these different sciences. And my area has mostly focused around what we call geomicrobiology, which is the interaction of microbial forms with the rock and mineral environment in which they live. So what is the real estate? This is kind of it. This is a beautiful picture of one of the most spectacular caves in the world that, luckily enough, was about an hour and a half drive to the entrance of the cave from my former university. And we were the science leads on the exploration of this cave. This is a cave that had been known for a very long time and actually trashed out and then rehabilitated by a local caving grotto. The avocational caving community provides thousands and thousands of hours every year of free labor to federally managed lands and is a major part of conserving these environments. And so the beautiful White's river that the young lady is standing on is known as Snowy River, and this is the single largest, by orders of magnitude, single largest cave decoration in the world. By cave decoration, we mean things like stalactites, stalagmites, but a lot more exotic stuff. And this goes on for 18.5 kilometers so far, and we are still exploring it. So anything in a rock fracture is really part of the same kind of subsurface microbial habitat. All rocks fracture at some level, and those fractures are living space for microbes. Those special parts of that rock fracture habitat that store water, very important to us as a species, known as aquifers. Those are even richer because they have the potential for transporting materials more freely. Caves and mines are significant because these are windows into this rock fracture habitat, and they're also even richer in niches because you have the interaction of both the gas phase, the water, and the solid rock material. And so you have just this amazing array of habitats for organisms to inhabit. This is not just true on the terrestrial land masses. Those are the areas that I work in. I primarily work in air-filled caves, although I've done some work in water-filled caves. But the ocean floor itself is highly fractured and completely infested with microorganisms down to at least a number of kilometers, maybe 5, maybe 10. We don't really know yet. So the message here is that the entire crust of Earth, down to 5 or 10 kilometers, is heavily colonized by indigenous, that means organisms that have always been there, life forms. And this is a hidden part of our biosphere that really we only began to have an emerging picture of over the last 20 years or so. So that beautiful sparkling white structure, I show it in this image here. You can see it on the right-hand side. And That's beautiful. But what we're really interested in is all that dark stuff that you saw in the previous picture, all that dark material that you may or may not have noticed on the rocks. That's where the microbial action is. That is completely deposited and created by the organisms that are busy eating the bedrock of that cave. They are not just hanging out there, they're munching down a lot of that material, and this makes them a very unique life form. They're geologically active, and they're doing all kinds of things to transform the planet. So one of the most fascinating things about the subsurface is just how different it is. This is one of my favorite examples. This comes not from my work, but from colleagues John and Susie Pint. Susie's Mexican, John is a British fellow, and they do most of their work in Saudi Arabia or Oman. You can see the background picture, of course, is the Rub al Khali, which is a very dry sand dune desert. Very famous area, the Empty Quarter. And within that area, geographically, there are many caves. And you can see Susie peering down on the left-hand side. She's peering down into a small opening. You can see the lower center picture shows someone rappelling, is rigged on the surface and rappelling down into a really big one. And when you get in there, you have literally gone to a different planet. You have gone to an area where there are diveable pools, where the biology that is inhabiting those pools are relics from perhaps 20 million years ago when this area began to become a desert. So these are time capsules, and it's such a different world. Even in the cases where you have these big, giant, gaping holes, you still have enough of a boundary condition to make the subsurface wildly different from the surface. There's some really obvious stuff about the subsurface. One is you don't have sunlight past that entry zone. So this means you don't have photosynthesis playing a big role in the food webs of caves, or at least not directly. So there is transport of organic material and nutrients from the surface biomass. But you have that stress on the environment, but you also have very benign conditions. So the fact that caves tend to be high humidity and very constant in temperature within a given cave, even though different caves have different temperatures, this is a very benign aspect of the environment. So organisms benefit from that. They pay the price when it comes to making a living and what they have for lunch. So those are the challenges for them. They're typically very low organic nutrients in these environments, but they're mineral rich. So the kinds of organisms that I have studied and I'm very interested in are ones that make their living not off organic material like we have for lunch, but off redox transformations or oxidation transformations of metals and other minerals in the environment, and they get tiny amounts of energy from that, small numbers of quanta of energy, and they couple that to carbon fixation, just like plants do with photon energy. So it's a similar process. They are making food, they are making more of themselves, but they do this without benefit of sunlight. One of the great things about caves is that what happens in the cave stays in the cave in many cases. So you don't have ordinary surface weather, and this means that a lot of these microbial processes, and in fact even macroscopic organisms, will self-fossilize in these highly mineralized environments. And so you can see the traces of even past life without the usual processes of life being buried and then replaced and fossilized in the way that we normally understand it. So the geomicrobiology itself is a very elaborate and complicated area of science, but really it boils down to this. What do microbes do when they interact with rocks and minerals? And the things that grab my attention the most is that they are master chemists and transformers of materials. So they destroy bedrock at a significant rate, and then they take that stuff that they munch from the bedrock or disaggregate from the bedrock, and they basically poop out unique biominerals. And you can detect those biominerals, and those biominerals are clearly, in many cases, a product of life. The textures and even the patterns of the precipitation of those minerals are an indication of life, or what we call a biosignature. So a biosignature can be anything as obvious as a big giant dinosaur bone, or it can be as subtle as isotopic shifts in the geochemistry of the environment. So there's a long list of cool stuff that the geomicrobes do. This is just a list here. They really are geological weathering agents. They probably are involved in a lot of low temperature economic minerals that we mine, so they have a lot of practical value. And they produce all these wonderful minerals, and I'm a mineral collector. I don't know if there's others of you in the audience who are, but a lot of those exotic minerals seem to be the product of microbial activity. They produce things that we can use that are completely underexploited at this point. So they are in an extreme competitive situation, and they are competing with each other for the limited amount of energy that's available in the environment. And this seems to cause them to make a very large array of different kinds of chemical compounds that they can use to keep each other at bay, okay? So it's almost like chemical warfare in some sense. And these have potential pharmaceutical potentials. There are many, many different antibiotics that they are producing that are completely unexploited. Certainly low temperature enzymes that can be used for industrial processes. On the more esoteric and strictly scientific side of it, we almost never find anything that is known to science. When we do the genetic analysis of these guys, almost everybody is novel. They are no more than maybe 90% congruent with other known life forms in the database. This is an unknown world. And there are implications for the very origins of life and its early evolution. Some people have actually suggested that the subsurface might be a more benign place to actually generate the prebiotic chemistry that you might need. Who knows? That's a very difficult thing to even study or think about. Well, when you wrap all this fabulous package up, of course, one of my main focuses has always been, and now is pretty exclusively, the astrobiology implications of a subsurface biosphere of such biological richness. How do we translate that into something that we can understand? This is one of the big challenges of astrobiology. We are really being asked to write a field guide to organisms that we don't even know exist. And that harks back to that cartoon that I showed you from the New Yorker, which is we have to think through a lot of this stuff from absolutely first principles, informed by what we can learn from the environments here and what our kind of life can actually do. How can our life push up against the envelope of these difficult conditions? So let me turn for a minute to the issue of caves. So I've mentioned caves and talked about the fact that I work in them. Why? Originally, it was because the environment was so entrancing and so chemically different in many cases, or different in terms of temperature or other features that were extreme, and that's what really drew us. But I have a background in planetary science too, and so it was immediately obvious to me that we should be thinking about where and how we would look for caves on other planets. This was at a time where no such thing had been actually really detected. So at first it was an exercise in complete speculation. But we thought about this deeply and how we would approach it. And what became clear was there was a sliding scale of what you could actually see. And in the early 2000s, we began to realize, as we sent missions back to Mars, and we had this orbital platform, this imaging capability once again, that we were seeing things that looked very much like volcanic caves, a byproduct of different volcanic processes. And now we know that there are tons of them on Mars and the Moon, and in fact other bodies as well. But there are first principles arguments here. So if you go down my little slider there in the direction of speculation, you have lots of ways of making cracks on a planet, both internal and external forces, internal seismic events that may occur on some bodies, maybe not on others, but every body in the solar system is being hammered by external impacts. And those external impacts create cracks, and when you have cracks, you essentially have caves. If you have fluid in the system of some sort, you have the potential for enlarging those, but you don't even need that. So there are lots of ways to produce holes in the crusts of planets. And if you really let your imagination reign, you can think about mechanisms that are maybe not represented here in the caves types that we have on Earth. Well, some of this is not really new. I mean, some of this we've known about for a long time, really from the early part of the space age. Ron Greeley, who unfortunately passed away a few years ago, was a very eminent elderly scientist at the time, and early on in his career, he grew up in the Pacific Northwest amongst lava tubes and volcanic features, and understood immediately that on the moon we were seeing features that were remarkably geomorphically similar to the kinds of things that he'd grown up on. Well, now our eyes have been opened, and I just put together a quick potpourri of different bodies where you can see features that appear to be lava tubes. You can see here the moon and Mars and even Io and even Mercury. Who knew that? And new processing of the old Venus data, radar data that now allows us to interpret better than we did at the time the data was collected. All of these bodies have clear indication of volcanic cavities of one kind or another. It's interesting that even though Ron Greeley and others were pointing to the similarities between the volcanic terrains that we have here on Earth, in the early days of lunar exploration, it was 40 years before actual caves were confirmed on the moon. That was in 2009. And this was because of the lack of missions. This was not because of the lack of thinking power. So I just want to show you one of these monsters. This is from the fabulous HiRISE camera that has been opening Mars to our inspection in a way that nothing else has. This is a really big feature. It's 100 meters across. And if you are not familiar with thinking about what 100 meters is, that's what it is. We're always looking for analogs here on Earth that we can tap into to get a feel for this. And so on Google Earth, wandering around one day, I found this sinkhole in Utah. So as we were doing, we've been doing research in Utah for many years and other environments. And so we drove out to it, pick up for scale. This is about a quarter of the diameter of that Martian feature that I showed you to give you a feel for what this looks like. Doing work in the Galapagos Islands a couple of years ago, this is about an 80 meter, so approaching the size. And if you can see on the far rim, there's little tiny people over there to give you a feel. Now, of course, this is not Mars. It's tropical Earth, so there's green scum all over everything. But you can imagine this on Mars without all of that vegetation. So the message really on Earth is that we have caves in just about every rock type. If you go to a lot of tourist caves, you come away with the idea that everything is made out of limestone. This is not the case. We have a lot of limestone on Earth, and we have a lot of water on Earth, and so we have a lot of limestone caves. But you can make caves with a variety of geological processes and all of these materials and more. So that got me thinking about first principles. This is the wordiest slide, and all you have to do is look at the pretty pictures on the left side, or the pretty colors rather on the left side, trying to get divorced from our earthly thinking about these kinds of features. I first published in 2004 a scheme for looking at what the fundamental physics and chemistry of the different kinds of ways to make caves would be. And this is a work in progress. We revisited it in 2012. We're revisiting it again now. And now what we're trying to do is actually look at what we know about the bodies in the solar system. And you can see I have added another column on the right where we're trying to say that these kinds of processes are possible on those bodies and give us a menu for what to look at. I want to turn now quickly to the icy satellites. From my cave perspective, these are not ocean worlds. They're planet-sized aqueous caves. They are roofed structures. They've got ice over them. And if you actually look at what you want to maybe look for there are a whole lot of features, not only the planet itself, but also features that may occur for which we actually have analogs here on Earth. I'm not going to go into detail here because I don't want to run totally out of time before I show you some more cute cave pictures. We have features like that here. These are three of these images. The two on the left and the upper right one are in association with the active volcano in Antarctica. These are fumarolic towers on the surface with caves underneath them. So this is a potential model for these icy worlds. The lower right is from Mount Rainier. There are fumarolic ice caves there, and in fact, my team is in the field right now. My colleagues on this expedition back to our sites there. This is a new National Geographic supported expedition of ours. So I want to show you just a couple of pictures of some of our greatest hits. That upper right-hand side is another image from that Mount Rainier site. This is significant in terms of astrobiological environments because it is very cold. It's about -3 degrees C. It has all sorts of gases that we find poisonous, but that the microbes living in there are actually using as energy sources. On the other end of the heat spectrum is the lower left. This is 40 to 60 degrees centigrade, something like 140 degrees Fahrenheit. And this is a very hot system where we also see that microbes rule the day. All of that orange stuff you see on the walls, all of those beautiful gigantic crystals contain pockets with trapped microorganisms in them that are still alive after tens of thousands of years. The upper left image shows one of our caves that we've been studying since 1998. This drips with sulfuric acid, and we have to go in all these protective suits and breathing gear and all of that, but it's one of the biologically richest caves in the world, and this is because stuff that we find poisonous is actually the base of the food chain. That hydrogen sulfide is feeding the bacteria. Bacteria are feeding everybody else. Everybody's happy. There's bats in there. Who knew that bats could tolerate this? They're much tougher than we are. So this has made me think that there are really two different kinds, at least, of biospheres possible. The one on the left is our kind. It's obvious, it's conspicuous, there's life all over the surface. You could pick up that signal of the chlorophyll spectroscopically, maybe even from another stellar system. We are an obvious type of biosphere. But Mars or Europa and the other icy moons may be very cryptic and may be very, very difficult to get our teeth into. Some of the things that we're looking at are macroscopic indications of microscopic organisms. So even though the microorganisms themselves are individually minuscule, when you put them all together and they have this wonderful ability to transform their environment, they produce spectacular stuff. They produce red towers packed with tulip-shaped microbes that coat themselves in iron. They transform copper from sulfides to sulfates and oxides and silicates and produce all of that blue stuff that you see dripping down in that particular cave in Sardinia in Italy. They produce obvious goo. So the snotites in the upper center are the favorite thing that I talk about with second graders, and I like them a lot too. Some of our stuffy colleagues once suggested that we not call them snotites, that we call them microbial veils, and I'm like, you got to call a spade, and you got to call snot, because it gives the right idea, and chemically, it's much more similar to snot. So all of these examples are there for us to see. The biodiversity is fantastic. I will not bore you with actual data. I will just show you a pretty picture, but I will tell you that we have hundreds of thousands of strains of organisms that are no more than 90% related to any other strain on Earth. This biodiversity far outstrips probably the entire biodiversity of the surface put together, and we've only just begun to scratch the subsurface, so to speak. You're looking at a large percentage of the world's community that do this kind of work standing here before you. There's not more than 20 of us distributed throughout the world. We're trying to change that. So any of you students who want to work in this stuff, there's a lot of low-hanging fruit. Energy-enriched environments. This is that sulfuric acid cave that I showed you. All of those organisms are living in there. The snotites are the most famous ones, but the stuff that I'm holding in my gloved hands, we call those phlegm ball mats. You may be picking up a theme here that when you're in a cave like this that's just dripping with life, the analogies to the human body or any body is really in there. Those live in anaerobic conditions. There are worms living in there in the complete absence of oxygen, so that's a highly evolved metazoan animal living in essentially zero oxygen. The fish are highly adapted to this environment that poison us. These are also vertebrates. 5 species of bats, including vampires, regularly live in here and in fact have babies in here, and how they do that under those circumstances, I don't know. So there are probably 30 or 40 PhD dissertations in that cave alone. We produced 3, so there's much to be done. This system has gained a huge amount of attention, and that is because of these giant crystals. It's been the subject of 2 National Geographic television specials, several different articles in National Geographic. Media people from all over the world have covered it incessantly. We were privileged to go head up 2 of those National Geographic expeditions. This was a rare opportunity because this system was only available to us because they were dewatering the entire system for mining. So these were caves, our caves, that have no natural opening. And we only were able to access them because the miners accidentally broke into them. They are in a very hot environment. So you see us in the little orange suits, you might think we're trying to stay warm. We're actually packed in ice in order to endure this environment. Even packed in ice, we can only stand it for about 30 minutes, and then the stress on the body starts to cook your brain, and your limbs become weak, and it really becomes life-threatening. And I learned that when I stayed in for 55 minutes because I was really trying to get this sample, and really, it nearly killed me. So I kind of learned my lesson, although, once again, I never retained that information for too long. So this is kind of the general scope. There are these giant selenite crystals. This is the same mineral as wallboard, intrinsically, chemically. It's not that exotic. But the form in which it finds itself is spectacular. And it's very hot. The water that was drained for mining has now reinvaded the cave. So this is no longer accessible. As of about 6 months ago, the mining company Penelope, which is one of the big Mexican mining companies, this is a commodity mine with zinc and copper and lead. And the commodity prices were no longer supporting the $7 million a year in pumping costs. So this is now flooded. This was a precious little snippet in time where we could actually study this system. But we have lots of samples. We have genetic analyses of a lot of the organisms in there. It's a saturated humidity, and as interesting as the crystals themselves are all that orange stuff on the walls, that stuff is alive. And so what first persuaded me to go into the system was the images that you see on the left, electron micrographs sent to me by an Italian colleague, Paolo Forti, who's one of the great cave mineralogists of the world. And he said, I think that these are microbial fossils. What do you think? And I'm like, oh, yes, they so are. We've seen this kind of stuff elsewhere. So I was sold, and I was expecting to find a lot of fossil material. What we found instead is that those orange walls are entirely encrusted with what you see on the right. Those filamentous little guys are diving in and out the iron oxides. They are very weird organisms. Their closest relatives are organisms from the Kamchatka Peninsula, a cave in Italy, and a cave in Australia. You explain that to me. I have a theory. So the results so far is we've done age dating with other colleagues. The big crystals themselves are about 500,000 years old. There are pockets within those. And within those pockets, we were able to, because we know the growth rate of the crystals now, we were able to determine how far in time we were reaching as we sampled within the crystal structure to get some of these. And that ranges from 10,000 to 50,000 years that the organisms trapped in these have been alive. That was the biggest surprise. We were actually really trying to just look for DNA that we could extract to see if it had still survived. And what we found was actually live guys. So this is, very exciting. We have not yet published this because I'm very cautious about these things, and we're doing a lot of additional work. There's a huge virus load in there. When you have a huge virus load, that says indigenous environment and indigenous community. Let me tell you one more case study, and then I'll wrap up. So the hunt for blue goo is an interesting story. It did not start in a cave or a mine. It actually started in the mineral cabinets of Harvard. So Carl Francis was the curator of the Mineral Museum at Harvard at the time, and we were on a completely unrelated field trip to go look at pegmatites, because I'm a mineral collector. And he was whining to me at dinner one evening about his copper sulfides. I mean, that's what you talk about and such things. And he was saying, God, you pull these out of the cabinets and we go at them with the toothbrushes and they're all beautiful and we put them back in the cabinets, and in about 2 years, they're all brown and ooky again. And he said, do you think that could be some kind of life? Could it be some kind of contamination? And I said, well, I have no idea, but, give me the pieces and we'll go look. And we looked, and sure enough, I mean, it's covered with all these bushes and all these organisms. And so that got us really started. And then once we started to work on that, we discovered that in a lot of the caves that we'd already been studying in Hawaii and Venezuela and Mexico and other places, that this stuff was actually going on in the natural subsurface. So this wasn't just Harvard, that it actually was a global phenomenon. So we've been studying these for a long time, since 2001. They grow really slowly. And even though we do genetic analysis on stuff, from my point of view, as someone who wants to see what kind of minerals, what kind of bedrock these guys eat and what kind of minerals they poop out, I really need to grow them. So a lot of my work is zookeeping function. So it took 30 months before we saw anything. I was sort of about to give up. And then about 4.5 years into the process, we began to see that they were indeed transforming materials. Now they're 15.5 years old, same plates, okay? That's a whole other story about how you keep microbes living in plates that long. You can see some of the electron images here of what they look like when they're growing. Okay, so that's the backstory. The real story is that we've been studying these a long time, and we use lots of techniques. And when we're getting ready to do electron microscopy with these guys, we dry them in air, we stick them in a vacuum oven, and then we put them in another vacuum system and coat them with gold and palladium so that they're electron dense. And then we throw them into the electron microscope and we zap them repeatedly with electron beams, high voltage electron beams. And I was looking at some of the older samples one day and thinking, oh, we need to do some more images because eventually we'll live long enough to publish this stuff. And so I pulled out some of them and I thought, God, they look fuzzy and brown. And I put them under the optical scope, and sure enough, they were fuzzy and brown. And I thought, oh, no, it can't be those guys growing up through. But it is. They're growing up through, after all that abuse, they're not dead like most microbes. They're growing back up off the stubs. And now they've done it 4 times. I thought it was an anomaly. We did the genetics on them. We know who they are. They're not related closely to anybody else. They're a type of fungus. They just look like ordinary fungus you'd see in your refrigerator, but they're not. Or maybe they are. I don't know. But there are organisms out there that you can't kill very easily. And this means that they are very robust within the geological setting. So we got guys that are trapped and living for at least tens of thousands of years. We got bugs that you can't kill off by a variety of means. This means to me that Earth's crust really contains not only unique organisms, but organisms that are uniquely tough and that can persist over very long periods of geological time. This has implications for Mars, of course, if you think about changes in obliquity cycles on Mars and obliquity summers and springs and things like that. So part of our practice in going to these extreme environments is really to practice for how we think about doing life detection and how we put those missions together. In our cave work, we're really dealing with something that is pretty alien, and I show you some examples of these. So far, all we can do is see them in electron microscope pictures. They look like little mesh stockings. We actually call them microchoia, because they look like the cholla cactus skeletons, except minuscule. These things are about 1/2 micrometer in diameter, a few tens of micrometers in length. We have chased them all over the world. They're in just about every subsurface environment, in just about every geochemistry, every lithology, and every temperature range, every altitude, every latitude. They're all over. Maybe they're coming to get us. I don't know, but they're really slow. So that's the good news. And we can't match them up with the genetics because I can't grow them. And when you do the genetics of an environmental sample, all you get is this giant dance card with a bunch of strain numbers. So we see them, but we have a logical disconnect about how to put those together with the genetic information. I've shown these to every kind of microbiologist and plant biologist and animal biologist I know. Nobody has a clue. So right here underneath our feet on Earth, we have essentially what amounts to aliens that we cannot easily figure out who they are. It's some kind of life form. We don't know. So I just want to end up with a couple of thoughts about the role of exploration and its danger and risk. We go into these dangerous environments not just so we can go into dangerous environments. It's not a extreme sports show. There really are fundamental scientific reasons for doing this. But the exploration itself and the fact that there is danger actually brings another flavor to the science that I think can be missing if everything is tremendously safe. The danger itself really focuses the mind, and we put up with all of these things that I show here. You know, we have to cope with atmospheres that we personally can't breathe, great heat or cold, you know, rocks are falling on you, who knows what, your gear is failing, everything is going to hell in a handbasket. You know, this is expeditions, okay? So that's annoying, and you want to minimize the danger. But in a way, it really does focus your mind. It really immerses you in the environment in a way that is very hard to do in another way. And this is something for us to be concerned about how do we bring that flavor of true human exploration as we try to develop robotic devices to actually help us, both here on Earth, but also in extraterrestrial environments. The exploration alone is not enough. You have to couple that then with the rigorous discipline of science, which is not an easy field, as a lot of you folks are finding out. When you put those together, this is where the understanding emerges, and this is really the quarry that we're after. So all of the work that we and other folks in our field of astrobiology do is really focused on this elucidation of those three questions that I posed at the beginning. What is the origin and evolution of life on a planet, in a planetary context? Who are these guys, and can we find them? Can we recognize them? And ultimately, what happens to life in a universe? What happens to life in our universe, in our galaxy? So with that, I'll end up with a bat. Okay, thank you guys.
Great talk. Great talk. So we have time for a few questions. If you have a question, please raise your hand, wait for the microphone, and then stand up, ask your question. Thank you.
Hello. So I don't know how easy it would be to quantify this, but when you look at the environments in the caves, would you say that there's a particularly higher or lower concentration of microorganisms there compared to other environments on Earth?
There's a higher diversity. The biomass depends on the energy sources. So in some caves, like the sulfuric acid cave, because it's got all that yummy hydrogen sulfide that everybody is grooving on, it's dripping. It's massively biomass rich. When you go into other caves where you don't have that extra enrichment, where they're either competing for tiny amounts of organics that's actually in the bedrock, or they're making their living transforming manganese or iron or something like that, then there's much less energy, so the total biomass is less. So the answer is yes and no, and it depends. Okay. Come on, you guys.
Penny, that was fascinating. Hi, I'm Amir. I have a question for you about the depth that to which the organisms will extend. The Earth heats up at about 25 degrees per kilometer in non-spreading zones, which means at 10 kilometers we're at 250 degrees Celsius. Way, way, way beyond what anything can live at. So the Earth is sort of naturally sterilizing itself. And there's also, of course, radiation damage. How do you account for this?
Well, I think that there are complexities. First of all, the geothermal gradient is not constant. So there are variabilities. So that has to be taken into account. Secondly, there are organisms that are known to be able to grow at 122, but it hasn't been pushed much beyond that. But a lot of that is actually operational. It's very difficult to work under very high pressures and high temperatures in a standard laboratory situation to actually test whether some of these organisms can tolerate high heat. There have been anecdotal reports of organisms living in and around the hydrothermal vent smokers at much higher temperatures. The issue is how do you actually demonstrate that in a rigorous fashion? That has not yet been done. There are probably things that get you before the heat. And one of those very important things is just the living space. So at some point, the lithostatic pressure overburden becomes so high that you essentially squeeze out all the living space. And so I suspect that in many cases that actually occurs before you get to the cooking temperatures. We don't really know very well what those cooking temperatures are. But there are geothermal highs and geothermal lows. And so I would expect that where there is much lower geothermal flux, that you would have the potential for organisms penetrating to a greater depth. But we know people are getting them out of 5 kilometers. So at least at that level, we know that this is a lot of the South African gold mine work. But there are cores that have been done deeper to 7 kilometers where there is still some microbial activity. So, exactly how deep that goes, we don't know. And it leads you to speculate, what happens on another planet when you have a lower gravitational constant. I would expect the potential depth of penetration just on the basis of the lithostatic pressure alone to actually maybe extend deeper into the crust. So it's a fascinating thing to contemplate, and we have way too little information to really give a definitive answer.
There are huge sampling problems because of contamination.
Oh, absolutely. Huge sampling problems, the gentleman just said. With drilling, there's huge sampling problems. This is why caves are so wonderful. And in fact, even mines, which I think of as anthropogenic caves, are wonderful because even though they're dirty environments and impacted by humans and their contaminated equipment, at least it gets you down far enough, into the system that you have at least reasonable control over drilling from that point. So it already gets you some significant depth within the crust, and then you can try to bring to bear better sampling controls to eliminate, or at least control or identify contamination than you have if you're drilling right from the top. Yeah.
Back here. Up here. What do you think the prospects are for your research influencing the search for life on Mars?
Well, I'm slugging it out. I don't know. There are a lot of contenders. I have to say that in terms of the Martian surface, it's a pretty unprepossessing surface, and as far as we know about it now. So, in my view, you've got a couple of places to look. On Mars, you can go in the subsurface either by accessing natural cavities or by drilling, which, only people who have never been on a drill rig think is easy. Or you can possibly go and interrogate these newly confirmed as liquid brine recurring slope linii, or you can focus on the poles, perhaps. So it's one of the contenders. For looking for ancient life, it's a different game. There's probably a lot more places that you can look. But if you're actually looking for living, or perhaps recently, whatever that means, deceased life, then I think that it's one of the leading contenders. I think that when you're looking at icy moons with liquid interiors, you're looking at a cave. So, you're looking at a cave situation. And so I think thinking about it in the framework of being a cave is just sort of a natural connection.
You mentioned different chemistries for energy sources, but is water still a necessity?
Yeah, water is a necessity in the organisms that we have studied. On the other hand, I don't know that we have the techniques or mentality to reframe the question, and not sure that anybody is really looking for life that doesn't have water, and how we would. So many of our techniques of analysis and detection are dependent on the fact that we're looking for the aqueous chemistry, and we're looking for things that we recognize. And if it was really different kind of life form, it would be very hard for us to detect it. So it's a very difficult question. I wouldn't rule out that there are potential life processes that might happen when you don't have water. It's just I don't know how to look for them or study them.
Thanks for a fascinating talk. I was just wondering, given your work on microbial resilience, do you think our planetary protection protocols are currently sufficient to stop Earth life from contaminating potential sources?
I've just come off serving for 9 years on the Planetary Protection Subcommittee, and I care about it a lot. And I think we need more work. I think that planetary protection is doing what it can with the cadre of tools currently at its disposal. But really there has been a lack of resources put into innovative methods of sterilization or other ways of controlling potential contamination. So I think it's an arena in which we have much to do. Do I think that it's adequate for some of our current mission purposes? Yes, I do, as long as the protocols are stringently applied. But it's not the end of the story. And it's been very frustrating to see that while other areas of research in astrobiology and elsewhere have been given the resources that they need to actually advance, that planetary protection has been the poor stepchild of this. But it is a critical need that we have. We must be assured that we're not just detecting Earth life when we are doing life detection, and then for missions like sample return, it's very critical that we not return anything even astronomically unlikely to create any kind of problem here. I mean, I think the risk is not zero, although it's very small, because pathogens and hosts tend to co-evolve, and so, but it's the only home planet we have. So it's an area of great concern to me, and it's an area of concern to astrobiology because we want the science to have the very highest fidelity that we can.
Patty, very, very cool talk. I think you've been sort of dancing around the subject that these are not isolated from the surface community, because it looked like an awful lot of pictures that people were breathing. So obviously the oxygen's getting in from the surface. And, I think much the same is said for the deep ocean, the organics. So it's not divorced from the photosynthetic world. So my question is, could you really have a cave ecosystem that's completely sealed off? And have you looked at transfer of microbes and so on back and forth to the surface?
We actually do have some systems that are completely anaerobic and at great depth. The reason I don't show pictures of them is that we have to use other techniques, and we're not in there. There are very high CO2 containing caves, for example. Now, the truth is that I think that it is impossible to make the argument that you have any environments on Earth that are truly completely uninfluenced by surface processes, because we've been thoroughly infested for 4 billion years or something like that, plus or minus a few hundred million, and it's like the planet is a product of this interaction of the geology and life. So we have no clean controls here. All we can do, I think, Lynn, is approach it with that in mind, that that there really are caveats in all of these systems. I actually think that you can run a system without that surface influence. It would be a very low biomass system, probably at a very slow pace of life, very much lower primary productivity than we're used to here. But not lower than we see in a lot of these environments. I think that some of the South African gold mine work is maybe the closest you can get. That's a fascinating study, whether the macroscopic organisms that they're finding, the worms, are really indigenous to that subsurface environment. I don't know.
Their ancestors were aerobic. So
it's right. I mean,
could you ever really have a primary system and therefore a real good analysis?
I don't know. I think it's impossible because of this. We are a hysteretic system. I mean, we can't escape the history of our planet, and we're trying to look for a completely sealed control. I just think it's ridiculous to claim that we're seeing it. I think all we're doing is trying to approximate that, and in the spirit of full disclosure, we have to point out all those caveats and then try to find ways to correct for them in our thinking. I mean, we're going to go look on Mars for these, right? And that's where we're going to find whether or not this is possible. And I think here, you don't want to overclaim the evidence.
Here, up here.
Hi.
Hi. Thank you very much for a very inspiring talk. You mentioned your efforts for DNA sequencing. Now, you didn't tell us much about the results. What did you find?
I only alluded to the results, which is we find tons of everything. And to give you the broad brush picture, almost nothing that we find, unless the cave is very, very shallow or has an active surface stream running through it, which occurs in Tennessee and other places. Almost nothing we find is very closely related to anything that we know. So we're looking at an almost entirely novel community or set of communities. The patterns that I think are the most interesting, that I hope that over the next century people will do more and more work on these subsurface phylogenetic relationships. What I actually think I'm seeing is the fact that we are looking at indigenous organisms and that their closest relatives are far flung around the globe. And what that says to me is that they are permanently in these chunks of rock, and that the transport mechanism is not the usual ways that we have on the surface by water and wind and so forth, or hitching a ride on an animal or something, that we are looking at the results of these chunks of real estate being dragged around by the, 100 million year, 500 million year supercontinental cycles. I actually think they're permanently in the subsurface. I think they get dragged around with chunks of continent. Some of them are lost by subduction, but enough remain that they are the founder stock so that they re-emerge evolutionarily when you get an intrusion into the subsurface, like rock fractures and caves and, in fact, human mining activities, all then allow an increasing niche richness to occur in those invasions into the subsurface, which allow those founder organisms to flower once again. This is the only uber hypothesis that makes sense to me. I don't expect to live long enough to see this tested rigorously, which is why it's so much fun to say it, because I don't ever have to suffer the consequences.
So with that, please join me in thanking Dr. Penny Boston.